Okay. Welcome to, let me get rid of this. Welcome to the Samuel Memorial Lectures, one of the biggest lecture in the department. And apparently I'm new here, so I do some homework to make sure how old this. Ever since I trace back, at least I can find out Bragg iPhone gave this lecture in 1999. And ever since, you probably see lots of famous names, probably all the top statistician in the world that's given this important lectures. And if you think about Charles McCullough and all the Rob Cipriani and all the good friends, it's all there. If you think about last few years, last year was by Han Huang, a year before was by Percy Diaconus and so forth. So those are, I just try to impress Elizabeth. This is a very outstanding group. And by that, I'm very, very proud to present this year. I call it year one after pandemic. Hopefully that's a long one. But we're very proud to Elizabeth to join us today. I'm going to leave a room for George and Min to do all the careful introduction. But I'm really, really excited about this year's program. And thank you so much for coming to join us to add into this. So the program is prepared mainly by Ming and Amen for the committee. The committee did an excellent job. I would say this is probably the best choice we can think of. A really good program. So by that, George, it's all yours.
Okay, can you hear me?
Yes, very well.
You can see me fine. In preparing these remarks, I was trying to think back about interactions with Myra and who she was as a person and as a colleague. So I'll tell you a little story. Many years ago, Myra and Steve decided to have a breakfast for people at Purdue who were visitors. Somehow or other, I was invited as a local to participate in this breakfast, which was very interesting. And it really struck me how exciting it was to be having breakfast in the Samuels house in West Lafayette with people virtually from all over the world. And I think Myra really appreciated that. And that's why she invited us and all those people to breakfast. Anyhow, as the time went on, often there's somebody in the crowd that is somewhat negatively oriented. Elizabeth, you've probably known people like that in our field. Anyhow, this person, rather than get mixed up with he said, she said, this is person A who started complaining that Purdue isn't such a great place. You don't have mountains, you don't have an ocean. And I was trying to figure out how to intercept this conversation. And I was going to say, well, we do have a river, but I declined. Somebody else who is not afraid of expressing an opinion. Elizabeth, you probably know people like that also. This person got very upset and said, you don't come to Purdue for the mountains and the ocean, you come for the people. It's the people at Purdue that are important. And this particular person had visited more than once, so it became, oops, this person came back. Anyhow, in thinking about that and your visit, even though we're welcoming you to do the talk by Zoom, however, I hope we can invite you back in person to meet with the really exciting people at Purdue, and I think you'd enjoy that interaction. So welcome.
Thank you. I too hope we can meet in person.
Okay, so it's my great pleasure to introduce our Myra Samuel Memorial Lecture today. So Dr. Stewart is a Bloomberg Professor of American Health in the Department of Mental Health at Johns Hopkins Bloomberg School of Public Health. And she has a joint appointments in Department of Biostatistics and the Department of Health Policy and Management. So Dr. Stewart got her PhD in statistics in 2004 from Harvard University, and she's a fellow of ASA and a fellow of AAAS. So Dr. Stewart, has extensive experience in developing methods for estimating causal effects and addressing the complications of missing data in experimental and non-experimental studies, particularly applied to mental health, public policy, and education. So her primary research interests include designs for estimating causal effects in non-experimental settings, methods to address and enhance the generalizability of randomized trials to target populations, and methods for policy evaluation. And she has received numerous research funding from NSF, NIH, the US Department of Education, and Patient-Centered Outcomes Research Institute. And with many of her awards, I will just mention, like, including the Mid-Career Award from the Health Policy Statistics from ASA, and the Gerard Cox Award from Applied Statistics, and Harvard Award for Excellence in Biostatistics, and also the Inaugural Society for Epidemiological Research, Marshall Jove Epidemiological Methods Award. Without further ado, I would like to hand it over to Armin to chair the seminar.
Thank you very much, Professor Zhang. So Professor Stewart, if you could go ahead and share your slides. Okay, thank you.
Great. Thank you so much. It's really nice to be here. It is true, I've never actually visited Purdue, so I haven't seen the campus or met, well, I've met some people from Purdue, certainly in other settings, but I do hope that I can actually go to West Lafayette at some point. And thank you so much for both this honor. It was really wonderful hearing a little bit about Myra. I've given a couple of sort of memorial lectures at other places, and it is in some ways one of my favorite parts of it is hearing a little bit about these amazing people and sort of a way to help remember and honor them. So it's really an honor to be here, both as you and we remember Myra Samuels, but then also to see that list of previous speakers. So thank you so much. And again, maybe we'll do it in person sometime too. So I'm gonna try, this might be a little bit of a whirlwind talk, but I think what I'm gonna do is really try to keep a sort of fairly high level. I have maybe 30, 35 minutes because I wanna leave some time at the end, and I know we have to end at around 11:20. So I'm just gonna really give kind of a high level overview. It's also maybe the end of the academic year, so people might not want a lot of equations and things anyway. And what I'm going to be talking about is this area that has become one of my main areas of research in the past 10 or so years, which is basically this question of sort of external validity or generalizability. And I'll give a little bit actually of a kind of personal story of how I got to this as part of this. And we might not get into all the weeds, but I think that will be okay. And my goal will be just to help expose you to this area of sort of applied statistics, which has relevance across biostatistics, but then also other fields like education and social sciences and other areas. Okay, so, oops. Sorry, I accidentally clicked in the middle of the slide, which I should not have done. So then it went to the middle. Okay, so let's start with sort of this context. What I'm going to start out with are these sort of motivating applied questions that we might be trying to answer. So a health insurer might be trying to decide whether or not to approve payment for a new treatment for back pain. We might be interested in predicting sort of overall population effects of a broad public health media campaign about car seats for small children. A physician practice might be deciding whether they should train their providers in some new intervention and whether that would be cost effective for their particular patients. Or, have to put in a little bit of COVID. So a healthcare system might be wanting to know if sort of giving convalescent plasma to all individuals who come in and are diagnosed with COVID-19 would be useful. What unites these, and sort of what my focus is on today, is an idea of population average treatment effects. So it sounds from Armand like, at least hopefully some of you have taken a class on causal inference. I'm not going to get into sort of the specifics of causal inference, but give sort of this perspective, where the idea here is that we're interested in estimating an average treatment effect across some well-defined population. So this is in some ways separate from personalized medicine and trying to say, what is the right treatment for this person. In contrast, these are sort of well across a well-defined population. Is this intervention sort of effective on average? As we'll see as I go along, that population might be fairly narrow. So it might be a school superintendent trying to decide if some intervention is effective, would be effective for their students. It might be quite broad. It might be the US Department of Education sort of trying to determine whether some intervention makes sense. Underlying this, and we'll see as we go along, is that there might be underlying treatment effect heterogeneity. So it may be that this intervention is stronger for some people, is better for some people than for others. But again, sort of what we're going to be talking about today is a setting where we're interested in this overall average across that well-defined population. And so what's really interesting is, in some ways this sounds very basic, but to this, until about 10 years ago or so, most of our interest in causal inference and sort of thinking about study designs was really focused on what I'll formalize later as internal validity, sort of how well can we estimate the treatment effect in the particular sort of sample that we have in front of us. There had been less attention to this question I'm focused on today, which is how well those results might carry over to some potentially somewhat different target population. So what I'm going to try to do in the next 30-ish minutes is try to just give you kind of an overview for sort of some of the newer methods and thinking about how we can be more careful about this question of external validity and how well the results from one study carry over to another population. I do want to acknowledge there's going to be lots of reasons why results from, say, a randomized trial might not generalize to a target population. That could include scale-up problems, or that like the intervention itself evolves or becomes different when you implement it at a broader scale. For now, today, I'm going to be focused on differences in impact that are due to differences between a trial sample and the population, in characteristics that moderate treatment effects. So we're going to be sort of trying to solve a piece of this challenge. Incidentally, I'm actually an author on a chapter in a book that just came out, or actually it's coming out in about a month, on sort of the scale-up problems more generally. And that book has papers from economists and sociologists and sort of a whole variety of fields trying to tackle the scale-up problem. But sort of the focus that I have is the statistical question around the sort of generalizing effects from a sample to a population. Some of you who know a little bit about this area might know that there's also this term transportability. I'm going to tend to use the term generalizability, but the sort of fundamental concepts are the same. And if we have time at the end, I'm happy to talk about just the sort of slight differences in those terms. I'll also make a connection, I think, especially for a statistics department. I don't know, some of you might work in non-probability samples and sort of survey designs. And this question of how well sort of non-formally random samples might inform about populations of interest also comes up in sort of surveys in general. The difference here is that I'm focused on causal effects rather than, say, population proportions or sort of things that a survey might also be more concerned about, like population prevalence or things of an outcome. And today I'm focused really on causal effects. Okay, so let's formalize a little bit what I mean by this. And this is my little personal aside. So my personal aside, and Armand and I share a grad school advisor, which was fun to hear. So what's relevant? Okay, so during graduate school, I focused primarily on non-experimental study design. So things like propensity score methods to estimate causal effects in non-experimental studies. After graduate school, I actually went to Mathematica Policy Research for a couple of years and was a researcher there, where I was involved in large-scale randomized trials of public policy interventions. So education, healthcare, labor, sort of different policy fields. And so that time really showed me the challenges in doing, like, yes, randomized experiments are wonderful, but they do have their challenges to implement. You know, in school settings, for example, there might be a quote national evaluation, but done using 40 schools from around the country. And this led me to really try to think carefully about what are the true trade-offs of these different study designs. So that then led me to work with Kosuke Imai and Gary King, who I'd known from graduate school, to try to formalize this a little. And this was a controversial and is still sometimes a controversial topic, but our fundamental point was that if you're interested in a population average treatment effect, a well-designed, sort of representative non-experimental study may be less biased than a small non-representative randomized trial. And that sort of our traditional ways of thinking about study designs and saying randomized trials are always better for causal inference don't necessarily hold if we, again, are sort of really interested in this population average treatment effect. So that was kind of our mindset. And so now I want to just formalize that a little bit for you. And that's what this paper does, is sort of talk that through. So what we laid out in the paper is that let's assume that we have interest in estimating, again, this population average treatment effect. So difference in potential outcomes if everyone in the population receives this treatment compared to if everyone in the population doesn't receive the treatment. So we have a population of size n, and it's just our estimand, the thing we want to estimate is this difference in potential outcomes under these two states of the world, with the treatment and without the treatment. So now let's assume I give you a data set. I haven't told you where the data set comes from, but I give you a data set and there's some treated people and some control people in it. And we might take an estimate of this population effect as just the simple difference in means between the treatment and control groups in this data set. And so that's what this D is meant to say. It's just sort of, again, in the data, we have an estimate of the PATE, but we don't know if it's a good estimate or not. So the bias in this estimate, I'm being a little loose with notation and expectations and things, but trying to just convey the intuition. The bias in that estimated treatment effect is going to be the difference between PATE and sort of this estimate we have. So then what we wanted to outline is how do different design elements affect the size of that bias. And so what we showed in the paper is that you can decompose that bias into 4 pieces. And those 4 pieces are these delta S terms, which, delta S terms and delta T terms, and then there's X's and there's U's. So the delta, let me go to the delta T terms first, actually. The delta T terms are what we sort of think of as internal validity bias. The bias due to the fact that the people who choose to take a treatment might be different from those who don't take that treatment. The X refers to things we observe and so can adjust for. The U refers to things that we don't observe and so can't directly adjust for. The delta S terms relate to this sort of external validity piece, this fact that in a given sample, our particular sample may or may not be representative of the population of actual interest. And so again, we show that under some simplifying situations, you can decompose this overall bias into these different pieces. That then allows us to think through, okay, what are these different trade-offs in study design? So just a formalization of that little intuition that I described earlier. And so what we lay out is that in an ideal world, if we want to estimate a population average treatment effect, we would randomly select people from the population and then randomly assign them to treatment and control status. And if we do that, then in expectation, our bias is going to be 0. I know of about 3 examples of that in practice, and most of them are sort of government programs where the government is able to mandate an evaluation and say, for example, the Head Start impact study, where Head Start centers were selected randomly, and then people who applied for a spot in those centers were randomly assigned to get in or not. Upward Bound evaluation is similar. These sort of, again, like large scale government programs implemented in centers and where they could randomly select the centers and then randomize people within that. But I think And honestly, I actually think we should do more of these experiments. For example, I think healthcare systems could use their electronic health record system to do this, but they don't necessarily do it very often. As a sort of aside, I'm partly just telling stories today. I gave a talk at Facebook on these sorts of things. And before I went to Facebook, I was like, well, why are they going to care? Because like they have their whole population and data set, like they could always randomly select and then randomize within that. But it turned out there were some nuances that that actually made the ideas relevant for them too. But I do like to push people to say, like, when you can randomly sample from your population, that's a great first step. But that's rare. So then what we sort of argue in this paper is that a typical experiment is one where, again, I do a lot of work in schools where you, like these large, quote, national evaluations will just, enroll 40 school districts from around the country. They will randomize within that. But we argued that that sort of typical experiment likely has big sample selection bias. And then, yeah, the treatment selection bias and expectation is zero. And so again, what we're really trying to get people to just think about is that a well-done non-experimental study where maybe we're using a large-scale, nationally representative survey, and then where we do a really good job of adjusting for confounding, that may in the end, I should have a total column on the right, that may in the end have less bias for our population average effect than this typical experiment. And again, we partly were just laying this out to kind of get people to think about this in slightly different ways than they had. It was pretty hard to get the paper published. I think especially people who come from a sort of clinical trials world, they don't really like hearing this message because they sort of think, oh, but my trial is perfect and great. And again, we were just trying to push that, especially in some settings, it may not be sort of exactly what we want it to be. Okay, I'll have one more slide and then I'll pause and see if there's any questions. So again, just to make some connections, and again, it's going to be a whirlwind today, but a lot of work sort of in causal inference addresses these delta T terms. So things like propensity score methods are trying to make these delta T terms as small as possible. What this new field of sort of generalizability is trying to address are these delta S terms and say, can we make these terms as small as possible in order to better estimate population effects? Okay, any questions? None in the chat box, any from YouTube?
Yeah, there's one question from YouTube. So it's about the random number generator. And the question is, which random number generator you would recommend to use to pick up from the population?
Oh, honestly, I have no opinion on that topic. I mean, I usually use R, and I don't have a specific one to recommend. I'm sure there are many, many that are all perfectly good. Thank you.
Okay, yeah, that's it.
Great.
Thank you.
All right, I will continue on and I'll aim to just kind of give a high, sort of summary of some of the big ideas. So the first question again is sort of how much do we need to worry about this? Because I just laid out this argument that, well, if we care about population effects, we should think about this. But there's very little empirical data to understand how big of a problem this might be. So first, I want to just describe that there has been work sort of formalizing when will there be this external validity bias? When will our sample estimate differ from the population effect? And you can show, and this is sort of intuitively and also formally, there will be external validity bias if sort of participation in the trial or sort of being in the study sample is associated with the size of impacts. So in particular, external validity bias is going to be a function of sort of the variation in probabilities of participating in a trial, the variation in treatment effects, and then the correlation between those two things. So just to say this another way, if there's no variation in treatment effects, if the effect for everyone is 2, then it doesn't matter who we enroll in our trial. Like the sample estimate's going to be the same as the population estimate. If there's no variation in the probability of being in the trial, or essentially if we have random sampling and can adjust for any probabilities of participation, there's not going to be any bias. So when there's going to be bias is when, for example, a trial enrolls more older people, and older people, are overrepresented in the trial. And so then the average impact we see in the trial might not be a good estimate of the average impact in the population. The problem is that it's really hard to estimate these quantities in practice because we don't actually observe these things in the population. If we did, we'd be done and we would just have the data we need, but we don't actually have direct data on them. So there has, and actually I'm gonna skip. So, well, there's been a tiny bit of work. So there have been a few examples where there's like a large scale trial and someone will subset that to sort of a smaller group of maybe a more typical trial sample. And they've shown that there can be bias that is about as large as sometimes the size of the effects that we are studying. So I think one direction this work is going is sort of even just starting to build a body of evidence of how big this external validity bias is. The other thing that we can do, though, is get evidence on sort of the pieces. So better understand why or sort of who does participate in these randomized trials. Honestly, it's usually not documented very well. Trying to change this in education research, but like people will just publish a study and say, oh, here's our 40 schools, without really any conversation about how they got there, how those schools relate to relevant populations. There's sort of an assumption that the result you see is going to carry over, but we don't know if that's true. The other piece is this treatment effect heterogeneity piece. And this is not what I'm focused on today, but there is growing methods research and empirical research trying to better understand effect heterogeneity, and that's really important. So I will just quickly mention, at least there is, I think, some increasing attention to potential lack of representativeness in trial samples. Sorry, it's sort of a busy slide, but I added to it yesterday because there was a new paper that came out this week. And so that's this Varma et al. Showed, for example, that older adults and Black patients are underrepresented in trials of cancer therapeutics. A few papers have shown that the people who participate in randomized trials of substance use treatment differ quite a bit from people seeking treatment for substance use nationwide. And then I did this work in education showing that districts that participate in these large scale evaluations are bigger, more urban, and lower performing than typical districts nationwide. And I want to say, we might purposely want to do randomized trials in those populations, in sort of these struggling school districts that sort of have a lot of need. The problem is that until recently, people weren't very explicit about that. And so then it sort of makes, and these evaluations get labeled as like a national evaluation. And so part of my interest is in just helping make sure people are sort of upfront about saying, well, here's what our sample looked like. And that way a superintendent in the middle of Kansas will sort of be able to take that into account and recognize that the participating schools might have been quite different from their own population. Any questions? All right, I will keep going. So this is gonna be whirlwind, but just to give you a sense. So now the question turns to, all right, we think this is a problem. How can we fix it? So as I mentioned, as statisticians, I think we probably would all agree that it's best to deal with these sorts of things at a design sort of upfront stage. So ideally, we might design our study to have random sampling, as I talked about earlier. It's great, but rare. There's also a whole field going back to like the 1960s and Mosteller and some giants in statistics on what's called purposive sampling, where the idea is to sort of just try to enroll kind of different types of, so some small rural school districts and some large urban school districts, and just sort of try to have sort of variation across the sample. A challenge with that I think it's good, but it doesn't have sort of the formal representativeness and unbiasedness that the random sampling does. And then in many fields, especially in mental health, where I do a lot of my work, there's been a push towards what are called practical or pragmatic clinical trials, where the idea, again, it's not sort of formal representativeness, but at least an effort to enroll a more broader set of patients, or use healthcare systems that are not just Hopkins and Harvard and Purdue, not just sort of like the really large universities, but also try to sort of go out into other areas and sort of try to get towards representativeness. But all of these basically, again, sort of say, well, let's just try to select subjects for our study in a particular way. I think there actually needs to be a lot more work on sort of how to do this well and feasibly. But then the question is, well, what if we already have studies done? Like, as statisticians, we also know that often people come to us and say, well, here's my data, like, what can I do? So some of you might be thinking, isn't meta-analysis, doesn't that deal with this? And the challenge is no, it doesn't, in the sense that meta-analysis basically says, okay, we have 10 randomized trials, we're going to pool them together in a meta-analysis way and come up with an overall estimate. And there's sort of a feeling, I think, that then that is more representative. But the challenge, of course, is that if all of those trials are conducted in the same non-representative populations, then this doesn't necessarily help. So it could help if the trials are like strategically done in different parts of the overall population, but it's not a guarantee. A former postdoc of mine, Huanyi Hong, it was great because she knew meta-analysis before working with me, and then we did generalizability stuff together. So she's now at Duke, and she's sort of trying to bridge these worlds. But sort of a default meta-analysis doesn't necessarily help. There's some really cool work that I would love to dig into more. I haven't had a data structure that allows it, where you can like combine multiple studies. So maybe we have a small non-representative trial and then also a large non-experimental data source, and sort of formally combine them and model the biases that there might be in both. So there's really nice work by Turner et al. And then Ellie Kaiser at Ohio State has done stuff. So I think that's potentially a really important direction. But what I want to talk about for the next 5 minutes is that we often, especially in the fields I work, we often just have one study. There's been like one trial conducted of some behavioral intervention, and we might want to get a sense of how well that result carries over to target populations. So there's, I could probably bucket these first two bullets here, but basically there's two strategies to then use data from the trial and some population information to predict impacts in the population. And at a basic level, you might be thinking, yeah, okay, like we could, the post-stratification, estimate if we know that age moderates treatment effects and the age distribution differs, we can estimate the age-specific effects and then reweight them using the population proportions. Easy, great. The problem is that often we have more than just one characteristic, and so you run into like small cell size problems. So there's been two main approaches that have been developed to try to do this. The first is sort of weighting-based approaches, which I'll illustrate really quickly, which basically model kind of like a propensity score or a survey sampling weight, basically model participation in the trial as a function of characteristics, and then use something like inverse probability weighting to weight those trial subjects to look like the population. The other approach, which also can work quite well, I'm just not going to illustrate it today, is basically fit a flexible model of the outcome in the trial sample and then use that to predict outcomes in the population. And so there have been some work showing that Bayesian additive regression trees work quite well for this, also TMLE. So I should say up front, all of these rely on having the individual level trial data and then having population data on characteristics. We don't have to have population data on treatment or outcomes. The assumption is that we don't have that yet. but we can use the covariates to sort of equate the trial sample towards the population. So let me really quickly just run through a quick example of this. So this is, it works great for sort of pedagogical purposes. This is a trial in HIV ages ago, which was looking at the effects of highly active antiretroviral therapy for HIV. And they randomized about 1,200 people and found a very strong effect, a hazard ratio of time to AIDS or death of 0.51. So very effective treatment. So what Steve Cole and I did was we sort of asked the question, well, what if the US could sort of give HAART to all newly infected people? What would be the overall population effects on time to AIDS or death? So what we did was we, from CDC, got the number of people newly infected with HIV in 2006 and the joint distribution of just a couple characteristics. It was all that we could get, but basically sex, race, and age. So we had the sort of US population by sex, race, and age. So this documents kind of what that looks like. So we can see the trial sample compared to the population tended to be sort of had more 30-ish year olds, much fewer young people under 29, more males in the trial, and more whites, fewer blacks. Importantly, and a big limitation is we don't observe CD4 cell count, and so we're not able to adjust for that, and that actually might be an important moderator. But for now, we'll adjust for the sex, age, race distribution. So what we do is we can basically, again, sort of fit a model of being in the trial as a function of age, sex, race, and their interactions, and then weight the trial subjects by their inverse probability of being in the trial. This is a sort of very basic version. There's refinements that could be made, but just to illustrate. And then we'll estimate effects using the trial data, but weighted to the population. And what's important in this context, and why this makes a nice pedagogical example, is that we saw that age, race, and just they should say sex are all significantly associated with membership in the trial. And I didn't show you, but these factors also moderate effects in the trial. And the largest effects were seen for those in their 30s, males, and Black individuals. So then we can do something like shown here, where we can, again, weight the trial to the population on these characteristics and get, in this case, like the age, sex, race weighted estimate, and say, if sort of using the trial, if the trial looked like the US population in terms of age, sex, and race, the hazard ratio would be 0.57. Ironically, it's actually remarkably close to 0.51. That's partly because the sort of effects cancel each other out in some sense. So the age weighting kind of makes the effect go down closer to 1, race moves it the other way, and then it kind of evens out. The interval is wider, though, you might notice, because we are now sort of extrapolating to the population. So we're paying sort of a variance price in trying to estimate this population effect. So I'm basically out of time, but let me just, because I do want to see if there's questions and discussion for a few minutes. So I just quickly want to say that the key assumption that has if we wanted to interpret this 0.57 as the population effect, we have to assume that there's no unobserved moderators that differ between the sample and population. And again, that's a big assumption, especially here, like CD4 cell counts is something that we know does moderate effects, and we're not able to adjust for that. So that's a limitation, and this is called this conditional sample ignorability. And so some of my other work then has turned to developing sensitivity analyses to say, well, how much would our impact estimates change if we tried to account for this moderator, and sort of make different assumptions about the distribution of CD4 counts in the population and see how that changes our population effect estimates. So I don't have time to go through that, which is fine. I'm happy to share these slides. So I just want to end, and again, just hopefully this has motivated you to sort of think more carefully about sort of how well results from trials generalize. Often, I'm doing some hand waving. often at the end of a paper, people will put some paragraphs that say, well, this study sample may not be representative, and they kind of have just some vague language about this. These methods can help quantify that and sort of put some statistical framework in how do we think about really sort of how well might the results generalize to target populations. So again, sort of a way to turn general concerns about external validity into at least more quantitative analyses. A challenge that I just want to flag is that data is a significant limitation here. Just like in that HIV example, when we didn't have CD4 cell count in the population, I have found that it's often very difficult to have a sort of robust set of factors that are observed consistently in a trial and a population. So my plea is that if you're involved in some studies like this, sort of think in advance what variables would be good to collect that will allow sort of comparison with the population. Like, are there variables collected in large administrative data sets where you might want to collect them in the same way in the trial so that you can use them for these sorts of adjustments? And I think that the data piece is a really important one that's often unaddressed. This is just a listing of some sort of more current work in this area. I've actually alluded to some of them already in terms of meta-analyses. Issa Dahabrat, actually, he just moved to Harvard. I forgot to update that. Has sort of done some really nice work on the sort of doubly robust estimators for this and some of the sort of more statistical pieces. But I want to close with this, and I think it's a nice lesson and reminder for statisticians overall, is again, sort of gets back to design. Like, if we want to be careful about external validity, we should design our studies to do that, either through careful sample selection or by collecting the data that's going to help us do this. So Don Rubin, my advisor, has this quote: with better data, fewer assumptions are needed. And then Leitzinger and Willett, who are some education methodologists, you can't fix by analysis what you bungled by design. And again, I think just a really important message for all of us as statisticians. And I think, you probably all know, you can't sort of magically fix a fundamentally problematic design. It's better to really plan for that up front. So thank you for patience with the whirlwind, and thanks to funders. I will leave this up for a minute. And again, I'm happy to share the slides if you want to see references, and happy to take, I guess we have like 5 minutes for any questions.
Well, first of all, Professor Stewart, I'd like to thank you very much for this extremely interesting talk. I'd like to add one other quote at the end. As Don said in 2008, for objective causal inference, design trumps analysis. So I think we're on the same page with you. I do have one question I'd like to ask, but I'd first like to see if anyone else has questions. And if not, I'm going to go to my question. So Professor Stewart, one question I had, as you're going through the analysis strategies for external validity, considering the moderators, I couldn't help but think of the method of synthetic controls, how that's gaining a lot of popularity these days. And I'm wondering whether that could have any potential at all for external validity, generating new synthetic data control and given the data you have.
Yeah, let me ask you what you mean by synthetic controls, because I have found that there are two main ways that term is used.
Yes.
One is in the interrupted time series world, and it's sort of waiting. Is that the one you're thinking of?
Yeah, so let's say you have longitudinal data and you care about external validity in that case. Is there any potential for using that?
I think so. I think so. A current postdoc of mine, Noah Griefer, is helping me learn a lot about sort of more sophisticated weighting approaches. And he's a big fan of this thing called energy balancing, which I'm just starting to learn about, where you can sort of have multiple constraints. So you could like try to match sort of longitudinally, like treatment to control, but also match to a population, right. And sort of try to like have balanced constraints basically that sort of do both of those things. So yeah, I think those sorts of things could be useful for those contexts. I think the problem in synthetic control world in general, though, is going to be that often those methods are used with one policy state. And so I think you sort of might have to ask the question of like, do I really, like, I could run the analyses, but do I really trust them?
Right. So we're going back to the same old ground. Gotcha.
Yeah.
I'll open up to further questions or comments.
Maybe I could ask a question. Let's see. Oops. So this is more a question of motivation. So you said you didn't really want to talk about in personalized treatment, but it seems to me that if the problem is a mismatch, in the population groups, isn't it also useful to just return estimates for each of these groups rather than just a single number for the whole population? Or is there any trouble with that? Yeah.
I'm so glad you asked that. There's no trouble with it. I think, and this comes up a lot because, right, it's like, well, if the treatment is better for older people than younger people, shouldn't we just give it to the older people? So this is one of my lessons, like sometimes like generalizability methods should not be applied all the time. Like it depends on your research question. And that's sort of why I started with my first slide, that these methods are going to be relevant and useful when interest really is in a population effect. And for example, a decision maker has to make a decision that cuts across their whole population. So cost effectiveness studies, I actually think probably should use these ideas more frequently. Or again, a lot of public health interventions where it's implemented at sort of at a scale where you kind of just give it to the whole community or you don't. And, vaccination programs or things like that. So you're totally right, though, that if underlying that, like, for example, so that's why we might want to do it. But you're right, like maybe the intervention is actually bad for some people, in which case we should acknowledge that and say, okay, yeah, you might want to give it to your whole patient population, but you really shouldn't. And so I think, like, again, it gets back to what is the research question? And the premise here for this work is that the research question is this population average effect. But certainly some studies are not going to have that as the research question.
Great, thank you.
Yeah, here's actually another question related with generalization. So the question is, has there been any work into how to generalize to a target population with no representation in the sample? For example, pregnant people are often excluded from the samples.
Such a good question. And in some ways, unfortunately, that's not really a statistical question. That has to get to sort of the science. So, and this actually gets back to my transportability reference. So what that's referring to, just to make it concrete, is like, so transportability, the term is used when your sample and population are sort of disjoint sets. So I've mostly been talking about a case where the sample is a subset of the population. But maybe this actually came up in a project I was involved in last week. Like, there's randomized trials happening in Europe. Do we use those when trying to learn about the United States? And so basically you don't have data to inform whether you can do that generalization. We don't have overlap. So you basically have to rely on scientific knowledge about sort of whether you can generalize, and like sort of does in the example from the question, does pregnancy matter? And I think like data is not, I mean, unless you had maybe other data sets that would help, but the current data at hand is not going to be informative. So you just have to be willing to make that sort of leap. That might be where sensitivity analyses could be useful, because you could say, well, if this and that, if pregnant women, blah, 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 and sort of probe how much it would matter. The other thing, I'm talking a long time, but the other thing that I think could be interesting that I've seen is like, let's say people over 85 were excluded from a trial. You might be able to use data from the people under 85 to understand whether there's any link between age, and then you might be more or less willing to make the jump to the older ages. Like if 50-year-olds and 80-year-olds have the same effects, you might be more willing to go above 85. But if there's huge variation across ages, you'd be less willing.
Thank you.
We do have one additional, I apologize, Professor Lin.
Yeah, Elizabeth, this is a wonderful talk. Thank you so much. I'm more on data collection people. So in my Deming lecture last year, I actually talked about for data collection, you should consider whether you should do it randomly or systematically. And I think either way, you need to design. Okay, now for systematic, it depends on problem. It would give a wonderful talk here. But even randomly, I think when I do the random number generator, there are different quality, different degree of randomization. So all random number are pseudo-random number. For pseudo-random number, some are more random than others. I'm not sure what I'm saying here, but just so in reality, when you use random number, we need some kind of measurement somehow, probably worth study to see how to quantify somehow measurement for how random is a random number in some way. And when you put into reality, then come to how sensitive is your random number. If some randomization actually doesn't matter, then probably that's not a big issue. But if quality of random number does play a role, that's probably related to your sensitivity, then random number, how random it is, might play a role there. So I just leave that. I don't have an answer for that, but I think just keep people aware that even random number, there's different degree of random numbers. Some are more random than others. But that's my 2 cents. Yeah.
Yeah. No, thank you. And I'm glad you're teaching about sample design and things, because honestly, some programs don't. So great to hear that you have class in that. My only additional quick comment on that is the additional challenge that comes up in a lot of these trials is that even if you pick randomly, and this is the problem with a lot of the large scale surveys, even if you pick randomly, people don't always agree to participate. And so there's sort of these huge biases that can come in based on sort of, we might do the best random sample we can, but then the people that we end up actually in our sample are often not exactly what we had hoped to have in the beginning. And especially like these large national surveys that sometimes have like a 3% response rate. So in that case, it's actually hard to know how beneficial the random sampling even was.
So thank you again, Professor Stewart. I think we have reached the end of today's session. On behalf of Purdue University's Department of Statistics, I'd like to congratulate you again on receiving the Myra Samuels Lectureship. And we all thank you very much for delivering your lecture today. Thank you again.
Thank you so much for the honor and for having me. It was a lot of fun.
Thank you so much. How you come to visit us in person at Purdue campus?
I hope so too.
All right.
Great.
Thank you.
Goodbye. Thank you so much.
Bye. Thank you.